Hello everyone, my name is Kyung-Won and I'm working at Theragen. Theragen is a genetic analysis company in Korea. Before the main talk, I would like to briefly talk about the interaction of genes and nutrients differs between individuals. Approximately 7 million years ago, human species obtained the food from animal hunting or gathering fruits. Recently, the modern human obtained food from diverse sources such as agriculture, hunting, and farming. Therefore, the modern humans are in the midst of natural selection to increase the adaptability to the environment by creating numerous DNA mutations in order to adapt to the plentiful food environment. The combination of genetic polymorphisms in each individual is very unique, and the response to nutritional components is also differently influenced to health disease results. Human ancestors need the genetic function of reprocessing the necessary nutrients and primarily processed and stored in the animal's body. After humans began to use fire, they began to eat grilled food, and the function of genes must have evolved from the ability to digest only raw meat to digest grilled food. Though the agricultural and the industrial revolution, humans had a lot of food to eat, and the genetic function had been evolving to efficiently store and utilize the food. Since the era when food became abundant, human genes have been spreading various genetic variant types to adapt to the food environment, interact with eating habits. According to the recent studies, there is clear tendency to consume very little of the recommended amount of healthy foods and excessive consumption of foods that require restriction. Unhealthy diets and malnutrition can often lead to diet-related non-communicable disease like diabetes. This study aimed to discover genetic markers that can affect the health improvement effect by recommending personalized nutrients to people on an unhealthy diet. The genetic information in my research was genome-wide SMP genotype data, and the information on diet is the intake of nutrients calculated in the food frequency questionnaire. Target health indicators or disease are metabolic syndrome-related clinical test indicators or metabolic disease case control. The meaning of the graph on the right is to analyze whether there is statistical significance by comparing the ratio of discriminated cases according to genotype. When a specific food or ingredient is divided into a group that eats a small amount, a group that eats of a moderate amount, and a group that eats a lot. As you can see in the graph, the GG genotype can be significantly increased from low consumption of the diet to high consumption of the diet. I would like to introduce my research results on the gene-diet interaction for obesity that I have been researching so far. The first study analyzed the interaction between genes and eating habits at the genome-wide level and discovered and reported a total of 4 genes related to abdominal obesity and obesity. The graphs on the right panel are called the signal plot, which are common methods of presenting results in GWAS study. A total of 4 genes affecting obesity according to gene and diet interaction were discovered. For example, in the case of CPQ gene mutation type, it was found that high vitamin C intake can increase the risk of abdominal obesity. It is difficult to talk about all other genes, but it has been confirmed that the mutation type of each gene and excessive intake of certain nutrients increases the risk of obesity or abdominal obesity. This is expected to be a good indicator for personalized management in clinics that manage obesity in the future. The second study used markers of well-known genes for dyslipidemia to investigate whether interactions with dietary habits could increase the risk of cardiovascular disease. According to analyzed genes, the correlation between protein intake and cardiovascular disease risk was confirmed according to the type of LIPC gene, which is a key gene for blood lipid metabolism. In the GG type, the prevalence of CCD was 1.32% in the normal protein intake group. However, the prevalence of CCD was 5.84% in the low protein intake group. In the GA type, 3.30% were in the normal protein intake group. However, the prevalence of CCD was 4.54% in the low protein intake group. The meaning of the result of this study is that through genetic marker testing among patients with hyperlipidemia, it is possible to select subjects for whom protein intake should be recommended to lower the risk of cardiovascular disease. The third study investigated whether the Mediterranean diet helps reduce obesity by using the markers of FTO and MC4R genes, which are representative of obesity genes. There was no particular tendency for the FTO gene, but in the case of the MC4R gene variant, TC or CC, it was confirmed that the obesity index significantly increased when the Mediterranean diet was not eaten. Thank you for your attention. If you are interested in the collaboration or genome analysis company, please see below. Thank you.